Okay, we're measuring out 500 milliliters of distilled water.
You want me to write on that?
Measuring out 7.5 grams of agar.
Thank you.
And 12.5 grams of LB media.
Okay, I'm adding the agar to 500 milliliters of the DI water.
I'm adding LB media to the water.
And I'm labeling it with a piece of tape as LB. And then we just cap it with a piece of aluminum foil.
Okay, so the media will now go into the Hansel and Gretel oven. Go ahead and
Can I just take it?
just put them in there. Get this. Hi. So I'm getting ready to pour my media plates. I'm labeling the bottom portion of the empty sterile plates, the dishes, with the media I'm using. In this case, it's LB. The media has been autoclaved, and it is not quite painful to hold onto, so it's ready to pour. If you wait too long, the gelatin will have set before it's ready to get into the dish. So you want to catch it when it's still hot. Turn on the flame. You use the flame to sort of sterilize the neck of the bottle between each stack. And you just like I did, just really quickly with the flame. Don't hold it there. You don't want the glass to get molten lava hot. Then I bring the stack of dishes. In my case, I'm using 5 dishes stacked on top of each other. You can use 4 stacked on top of each other. And I bring it close to the edge of the counter, and that's so that I can bring the bottom of the flask underneath the height of the dish, so I'm not dumping the media from a great height. This helps to prevent splashing. I lift the lid off of the bottom dish with the stack on top. I pour until the bottom of the dish is covered, and then I replace the lid and move my way up the stack, pouring smoothly. You want to kind of fill each dish with about 22 milliliters of media. That means you want to cover the dish before you stop pouring. And then when that stack is done, I flame the neck. I gently slide the still liquid media out of the way and move on to the next stack. If you get bubbles onto the surface of your plate, these can be distracting when you want to count bacteria or yeast colonies. So the trick there is to either not agitate the solution, avoid bubbles from the beginning. But if you have some bubbles, you can take your flame, open up the lid, and the flame will pop the bubbles before the gelatin has a chance to set. Especially when you get towards the end of your solution, you want to work perhaps more quickly because the solution is cooling more rapidly as you have less of it in there. And the goal is to get all of it poured before it starts to set. If you don't have enough to fill all the dishes, don't worry about it. As soon as the flask is empty, you come over to the sink, flush out the remaining gelatin residue with a lot of water so it doesn't set and become a great big cleaning problem. And you can leave the flask filled with water, set them off to the side to be cleaned up later. And that's it.
Right now what I'm going to do is pour plates. So I have a stack of 4 plates labeled with the type of media that I'm using, which is YPT in this case. And I have my media. So what I'm going to do is take off the foil and quickly pass the top of the flask over the flame to sterilize it. And then what I'm going to do is lower the flask so that the bottom of it is below the stack of plates. And then I'm going to pick up all of the plates, including the lid of the bottom plate, and slowly pour the media into the plate and keep pouring until the plate is filled, and stop. If there are a lot of bubbles on your media, as there are in my case, what I'm going to do is slide the plate over and pass the flame over the top of the media to pop the bubbles. And then I'm going to cover the plate and move on to the next one and sterilize the flask. And stop once the plate is filled. And then carefully slide the plates over, and then get the next stack and keep pouring. Once again, I got a lot of bubbles, so I'm going to pass the flame over the plate to pop them.